Hello and welcome to Principle of Cognitive Neuroscience Part A. As you may know, this course is divided in three parts. In main modules, I will train you to think about how the brain works and expose to you the main theory explaining its functioning. In main concept, we will tackle what constitutes a result in neuroscience and how can you really report it. And during the practicals, I will explain to you how to do things practically, put your hands on and actually do the analysis together. But without any further ado, we will start with the course of today, which is about epistemology, which is a complex word and also complex concept to tackle. And I thought like there is no way I can present to you different models of the functioning of the brain without, in the end, really giving you the master key to understand that the way we define our understanding of the functioning of the brain is highly biased by the way we can observe it. So, what is epistemology exactly? So epistemology is derived from the combination of two old words, epistemo, which means knowledge, and logis, that means reason. And the idea behind it is that with epistemology, we will try to understand the nature, the origin, and the limits of human knowledge. And well, you know, when you think about it, what exactly is knowledge? Well, you know, it's been defined as by Bacon in the 15th century, explaining what is a scientific approach to build knowledge. It is relying on observation to elaborate theories and models of the nature and the behavior of natural things. And that's knowledge. Without this, it is only belief. Do you see what I mean? So knowledge is really based on science and rely on the observation to elaborate theories and models of the nature and the behavior of natural things. We all do that to build science and make discoveries. And let me insist on this. So this knowledge is actually built incrementally through this virtuous cycle between theory, methods, and observation. In the previous course, I exposed to you the case of Phineas Gage, who had a radical change of personality after the passage of a metallic bar through his frontal lobe. The case of Tan, who was not able to speak anymore after the damage of the inferior part of his frontal lobe. And the case of Henri Mollison, who was not able to build any more declarative memories through episodic memories, through the damage of his medial, after the damage of his medial temporal lobe bilaterally. And those three cases are behind our beliefs that the medial frontal lobe is a key to the estimation of values and the personality. Inferior frontal lobe is the main region for the production of language, and the hippocampus and medial temporal lobe are key structure to episodic memory. But now, if you've seen the other lectures, you know that the brain is not made only of its surface. It is actually made of brain regions that are highly interconnected, tightly connected by white matter pathways. And still through the same logic of building knowledge, you have the observation and the theory, and through the method, you can actually refine or change the angle of the observation that you have onto something. It's like when people started looking at the universe, and they like stop looking with their eyes and look with instrument to look at radio wave, for example, and discovered like a new way of looking at things and understanding things. At the same way, you change your method, you sharpen or change your observation, and this helps you to elaborate new theories or make your theories evolve. And this way, we've been able to explore our three cases and see that if you look at the connections that were interrupted in those three cases, you can also build a theory that makes sense. For Phineas Gage, for example, the bars that went through his frontal lobe disconnected the amygdala from the rest of the frontal lobe, the amygdala being a key structure for emotion. If you prevent the frontal lobe inhibiting the emotion of the amygdala, you'll have easily emotional outbursts. We can see that, for example, for the cat. And the same bars being disconnected within the frontal lobe, exchange between regions will be a good explanation why Phineas Gage had difficulties demonstrating good planning behavior, such as going outside with thin boots and pajamas when it was raining and cold. In the case of Le Boyne, the damage is lesions that have never been studied other than from his surface organization. If you do an MRI and if you slice the brain, you see that the white matter is very much damaged in this case, and the lesion damaged the arcade fasciculus, which we know is key to language, but also the frontal ascending tract that later on we demonstrated is very important for fluency. And in the case of Henry Molaison, the lesion of the medial temporal lobe not only damaged the hippocampus, but also removed bilaterally the amygdala and the medial temporal cortex. And by doing so, it also disconnected the uncinate fasciculus, which is very important also to elaborate emotional memories. And we all know that emotions are a key important factor in the way we remember things. The fornix, which we know when it is disconnected after a surgery of cyst in the colloid, will lead to episodic memory disorders. And the cingulum in the blue, which is very important as it links the hippocampus with the retrospinal cortex and the entire cingulate, key structure of the default mode network, and also key structures that are impaired in Alzheimer's disease. And we all know that one of the key behavioral, like cognitive problem in Alzheimer's disease is the difficulty to build new episodic memories. And then, we went a lot further in the concept, saying that, the function might not be related only into the disconnection of the white matter, but maybe you have a brain with regions that interact even more than we think, and like the function just emerge from this interaction. And in that case, the more you interrupt the circuitry that is supporting this interaction, the less you will be able to do the function. And you have the illustration here with Phineas Gage, a red bar that is going through the frontal lobe, And this heat map that indicates the probability of disconnection of brain regions, and you have it side by side with activation in the brain related to tasks involving decision making or the manipulation of emotion. And you can see that this network of functional activations in healthy controls is completely disrupted by the bar, like by the disconnection produced by the bar in this case, completely explaining why this patient should have these symptoms. So changing the vision of people and giving them a new way to observe things is a good strategy to build on new theories, but never forget that you're going to make it available. You gotta get people on the train with you, otherwise you will be, just an outlier. So it's very important to engage as much as you can with open science to give the opportunity to people to test your ideas, and eventually take the risk to disprove it. But if you want to be a good researcher, you're in the game for making discoveries that last, not just strategically pushing out ideas and like not accepting that you might have been wrong. So to do so, we produce a software to allow people to modify their observation of brain lesions, which is a BCB toolkit. You can download it on our website. This software will allow you to study the tracks that are classically disconnected, exactly like the case of Phineas gauge and the frontal disconnection or the uncinate disconnection. You can do that with the Tractotron tool, just by putting the lesion inside the software and running probabilistic analysis. You just need a drawing of the lesion in the reference space that we call MNI. You can create maps of disconnection, which is more like the holistic vision, where you will look at the probability of disconnection on the surface of the brain and relate it with functional activations. And we provide a way to register the brain to the MNI space that we call normalization, just because people ask us for it. But we also provided new way to observe brain disconnection, such as cortical thickness as an area that is disconnected from other areas. Will show a reduce in thickness that is due to the reduce in the size of the neurons inside the cortex, or like the death of the neuron inside the cortex. As we know, a neuron that is disconnected reduces size or dies, and that's called apoptosis. Functional connectivity, I will briefly touch on this, which is another way to assess the way regions communicate together through shared variance in the signal, and you need a resting state functional connectivity measurement to do that. And then ANACOM 2, which is just a statistical approach, a non-parametric statistical approach for you to assess in small group of patients, otherwise you have like a wealth of other methods to assess like the contribution of cluster of disconnection or the contribution of cluster of lesions to your behavior. Okay. And as a demonstration of what you can do with this software to elaborate new knowledge, we applied it to a task, which is category fluency, and in patients with brain lesions, to try to identify the network related to deficit in category fluency. For those who don't know about it, category fluency is a neuropsychological test in which we ask participants to say as quick as possible as many words of the same category, typically animals, And they usually have 1 or 2 minutes to do it, and you count the number of words and you write down the number of words to see whether they have strategies. LC participants usually give a list that is following a little bit of a story, something like traveling in their head. Here you have chicken, horse, cow, donkey, earthworm, fish, dog, monkey. Well, this is not a good example. The one in the normal control in B is the best example. I think you have dog, cat, parrot, rabbit, snake, ox, deer, chicken, pig, cock, alligator, horse, monkey, duck, jaguar. So you can see how the healthy control started his little story from what can I find as an animal at home, and then he was like going through the forest with the snake, the ox, and the deer, and then go to the farm with the chicken, the pig, and the cock, and then went a little bit in his head in the zoo and found the alligator, the horse, the monkey, the duck, and the jaguar. So if you compare that with a middle cognitive impairment, at least some of them, you'll see that they have still this production of like kind of a comparable production in terms of number of animals they can produce, but the story is completely scrambled up. It's kind of like they're giving it randomly. So that will mean that, you know, sort of the way you can order your thoughts and you travel into your mind to explore those different animals to perform the best at the task of category fluency is all scrambled. And in Alzheimer's disease, which sometimes is a more advanced version of MCI, middle cognitive impairment, you have less production, the recollection of those different animals, the access to the category of the examples of the categories here is lost, and the prediction in Alzheimer's disease is mostly reduced. And because you can have dissociations in the impairment that you can have behind the performance in category fluency, that means that category fluency emerge from the interaction between different functions. In other words, category fluency is not really a function in itself. It's a combination of other functions, which is the exploration of the semantic network to assess whether you are in the same or a different category, the organization of your thought, like you're traveling into different place and maintaining what you already said, which is an executive function, and the production of these different words, which is made through language. And the three together integrate to produce this function that we call category fluency. I'll just take a minute to show you the picture of Chris Poulon, who developed the software and has been doing this research on category fluency during his PhD with me. So here's a roadmap of the analysis we will do to change the way we observe the impact of brain lesions and the interpretation that we can have onto what could be the origin of this disorder, which will be a decrease in category fluency. So first, we'll look at white matter track disconnections. To answer the question, what is the relationship between category fluency performance and the disconnection of well-known white matter tracks? Then we will try to go beyond classical track nomenclature. We'll look at the direct disconnection of brain areas to show the structural connectivity network or structural disconnectivity network. To do that, we'll produce map of disconnection and studies their relationship with category fluency performance in patients with brain lesions. Then we'll explore whether this network of areas that when is disconnected leads to a deficit in category fluency performance is a match with the area that are classically activated during fMRI for tasks involving the fluency, the production of language, and the manipulation of categories. And for that, we'll use a meta-analysis of fMRI data. Then we'll explore whether these regions that we have classically work together in healthy controls, even when you're not doing this task. And to do that, we'll look at the functional connectivity network to see whether those regions are classically synchronized together, and are we touching different network or networks that belong to the same large network of functional connectivity. And to finish, we will investigate whether for real, when you have a disconnection within this large network, you have a decrease of the cortical thickness. Well, you have a reduction of the size of the neuron or the death of the neuron that will lead to a reduce, like a decrease of cortical thickness that will be statistically linked to the fluency performance, category fluency performance. And without further ado, we'll start with the first thing, which is the white matter disconnections. So we had 60 patients with lesion in the right or on the left hemisphere. We look at the relationship of the localization of lesion with category fluency. Nothing came out as significant, so we started investigating patterns of disconnections. If we do the classical track analysis, here are like the tracks that we investigated. And we compare brain disconnection. Here you can see on the right bar chart. When the track is disconnected, it's in dark gray. When the track is not disconnected, it is in light gray. And when we look at the performance of the patient when they are disconnected or when they're not disconnected, in the left hemisphere on the left, on the right hemisphere on the right, it doesn't really matter. They will always have a performance that is below the performance of healthy controls. That's right. So you have a lesion in your brain, most generally, and here we studied mostly the frontal lobe. You will always have a category fluency performance that will be lower than the one of healthy controls. And we can see that because controls here in green are not ever crossing the standard, like the confidence interval that we can see as a little line of the patients, whether they have a disconnection of the track or not. But among the disconnections that are possible in the frontal lobe, some are worse than others. And here we can clearly see that disconnection of the arcuate fasciculus, whether this is the anterior of the long segment, the frontal ascending tract, or the frontal inferior longitudinal or frontal superior longitudinal, which are frontal connections, in the left hemisphere, will all contribute more to a decrease of performance in category fluency than any other disconnection. And that is really important because that shows that you have some specialization of specific track in the left hemisphere that are showing a relationship with a decrease of category fluency performance when they are disconnected. The same disconnection in the right hemisphere didn't seem to have a comparable effect. Now, this is a classical associationist approach, and the interpretation will be that you need those connections to convey information from A to B, B to C, to produce a category fluency. But we can also investigate the pattern of disconnection viewed from the surface by using the disconnector maps. And if you do so, then you use every lesion, produce a disconnector map like in Phineas Gage, and run statistics on it. You can see that some regions in the brain, when they are disconnected, contribute significantly to a decreased performance in category fluency. And those regions involve the middle part of the caudate, the entire portion of the thalamus and the ventral portion of the thalamus, the dorsal portion of the putamen, and those are subcortical structures. But on the surface of the brain, you also have in the left hemisphere, the pre-SMA, middle frontal gyrus, and pars opercularis, with a little bit of contribution of the intraparietal sulcus. And in the medial view, you'll have a contribution of the cingulate gyrus, and well, you have like the other side of the pre-SMA here. And you can also find some areas that contribute significantly in the right hemisphere, where you can clearly see that the middle frontal gyrus, the pre-SMA, and the insula were also contributing in the right hemisphere to the category fluency, and same for medial regions of the cortex. Now, the big question, so we can see also on the right on the chart that most of these regions were showing a significant effect and were significantly different between when they are connected and when they're not disconnected in patients. What we can ask after that is whether this network of region, which when they are disconnected contribute to the category fluency performance decrease, whether those regions belong to networks that is classically activated in inertia control during the production of fluency, or the manipulation of categories. And so then my next slide is about a meta-analysis of functional MRI activation in healthy controls during the production of fluency or the manipulation of categories. So we're going to do it in 2 times. I want you to pay particularly attention to the subcortical regions on the top left, the caudate, the putamen, and the thalamus. And what we can see is that the regions that were disconnected in patients leading to a decrease of performance in category fluency compared to healthy controls are classically activated with functional MRI during tasks involving the production of language fluency and the manipulation of categories. And now we're going to look at the surface of the brain. Bear in mind that I only had 60 lesions, so I didn't have a full coverage of the brain. So what we're going to look at is whether the regions that are classically, well, that are disconnected in the next slide still fall into the network of regions that are classically activated during fluency or task manipulating categories in healthy controls. And that's this next slide. And you can see that these regions mostly fall into this network of activations. So the regions that are disconnected fall into the network of activation in RC controls. And that will mean that somehow you need the interaction between these regions to elaborate the category fluency, and the disconnection of the circuitry behind it will prevent the achievement of this function. Now, we have a lot of regions here, and like the next steps that we wanted to do was to try to see whether these regions belong to the same network. To do so, we used resting state functional MRI, and then when you use resting state functional MRI, what you can do is you can look at slow fluctuation of the board signal in the brain. This is real-time fluctuation of the board signal in the brain, and try to build a statistical model that will segregate region according to whether they are synchronized or not synchronized in this fluctuation. This is particularly interesting because the way these regions synchronize together appear to match very well the way they activate in healthy controls. So you can see here that you have a network of regions that you can find with resting state networks that will match the social theory of mind, another network of regions that will match the production of language, another network that will match the relational effect, and another one that will match the motor activations. And even more exciting, this network are actually a match at the single subject level and can allow us to really harvest what is the specific pattern of activation of RC controls, leading us in the future to a better precision medicine. Now, if you apply that to the context of our patients, if you use all those clusters that were significantly disconnected and leading to a decrease of performance in category fluency, and run functional connectivity, you have a lot of functional connectivity map, and you look at similarities across those functional connectivity map, you end up with models that will involve 3 main maps that will explain 92% of the differences across the map of functional connectivity. And those maps are mostly the singulo-opercular network that will connect the cingulum with the operculum. The corticostriatal network that will connect the cortex with the striatum, and the ventral frontoparietal-temporal network that we can imagine is mostly supported by the arcade system. And those networks are the networks that are classically synchronized in healthy controls. What we can do, and we had the chance to have the resting state of our patients, is try to look whether these networks were functioning properly in our patients. And so we looked at the singulo-opercular, the corticostriatal, and the ventral frontoparietal-temporal network that you can see here at the bottom right of our slide. And you can see that 2 of those networks were showing a decreased functional connectivity, the corticostriatal and the ventral frontoparietal network, when they were disconnected in our patients, were showing a significant decrease of functional connectivity compared to when they were not disconnected. Interestingly, if you look at the singulo-opercular, you can see that there was no significant difference, and that's because in the spare patients, they were like this network was highly variable, and this did not allow us to reveal significance, and we just don't know why. Maybe mechanism of recovery or I don't know. It just like didn't come out significant. What's important to see here is that like if you test your idea or your model of functioning of the brain as an holistic perspective, here really disconnection is showing a decrease in the overall communication within the network in our patients, and this decrease of communication within the network should lead to decrease in size in the neurons or the death of the neuron in the regions, which will subsequently lead to reduced performance in category fluency. And this is exactly what we tested in the next slide. So we elaborated, we modified a classical method to measure cortical thickness so that it is applicable to patients with brain lesions. So we used advanced normalization tools that give you an assessment of a way to measure the cortical thickness. And usually it is applied to healthy controls with high resolution. We had the chance to have a high resolution T1 MRI in patients with brain lesions. You can see at the top the MRI, and there's clearly a lesion in there. The problem is the lesions can make the software crash because the software is really defined to work on healthy controls only. So what we do, we draw the lesion of the patients, and you'll draw some lesion of patients next Monday or last Monday if you watch this video later. And what we do, we're gonna take the information of the symmetric information of the opposite hemisphere and feel the regions that was damaged with the information of the opposite hemisphere to fake a healthy brain. And you can see here, and this is called an antimorphic transformation. It's been published in 2007 by Pashkev-Natchev, and this method allows you to hide the lesion and run classical analysis of cortical thickness on it that you can see at the bottom row. And what you just do at the end is remove the regions that were fake and replace it with this hole that you can see. And you have a perfectly consistent measurement of the cortical thickness in your patients. So what we try to assess is whether within the network that I showed to you, within the regions that were disconnected, we had a relationship with the performance of category fluency and the cortical thickness of those regions. And this is what we found at the end, which is, you know, with the disconnection, the brain, the neuron decrease the size or die, and the cortex is thinning within the network that is disconnected with functional connectivity. And the thinner it is, the lower will be your performance in category fluency, demonstrating that it is the integration within this large network that is the best predictive of the disruption of the integration within this network that is the best predictive of the category fluency performance from this, really from this perspective. So in sum, we played around with method to change our observation, and every time by doing so, we're touching different concepts a little bit of how the brain works. So like for category fluency, for example, I didn't present it here because there's absolutely no results. When you do classical VLSM, voxel-based lesion mapping, where you look at the localization of the lesions that will be related to impairment, you never really find any regions that will be significantly responsible when damaged with category fluency. But if you look at the disconnection of white matter tracks, you do find some. And I mean, like, okay, so I need to have afferents and efferents from regions that communicate together in order to produce this category fluency performance. But even though the level of disconnection of the cortex actually match the classical activations in healthy controls, this level of disconnection of regions that are significantly related to a decreased performance of category fluency match meta-analytic fMRI map in healthy controls for tasks involving fluency or the manipulation of categories. And these disconnections belong to regions that usually speak together, they synchronize their activity. We've seen that in healthy controls. And if you disconnect the circuitry between the synchronization of activities, the synchronization between these areas decrease in the entire network. And then this decrease of synchronization in the entire network lead you to have neurons that work less, they reduce their size, or they just die from isolation. This is apoptosis. And this reduces the cortical thickness in the regions that are disconnected. Finally, this reduced cortical thickness in the regions that are disconnected leads to a reduced performance in category fluency. And so, in sum, through the manipulation of method, we change observation, and the theory of how the brain works change a lot. Okay, and every time we can modify our vision, the light in the cycle is taking a different angle. But we can also, you know, we can apply this to even like deeper questions. Like you might know about Descartes' mind-body dualism, whereby Descartes was suggesting that, you know, some area in the brain got a direct connection with the spirit and the mind that will observe what's happening in the body, and to us scientists, at least me, I do think the mind is really ascribed inside the brain, its neuron and its interconnections, and the mind works directly with the body. There is no dualism, they are actually together. Is that true? Well, one syndrome, one neuropsychological syndrome, seems to show a dissociation between the two. And this is called Anazognosia for Hemiplegia. And that's patients that have, after a stroke, an hemiplegia, they cannot move their arm, for example, their left arm, but they are not aware of the loss of motricity in their arm. So they don't have the gnosia, the knowledge of the motricity of their arm. So what we tried to do, and this was with Valentina Pasella during her PhD, is try to investigate what will be the anatomical basis of this knowledge of your own body by studying patients with Anazognosia for Hemiplegia in opposition to patients without Anazognosia for Hemiplegia. So there were 95 patients against 79. And we did this comparison removing from the analysis confounding factors such as how old are those patients, their level of education, because this is important for them to understand their symptoms, the interval between their lesion and the moment we measured them, because some of them recover, some others do not, the size of their lesion, because bigger lesions are like usually producing more impairment than smaller one. The personal neglect, because you can have any neglect of your own body, the extrapersonal neglect, because you can have neglect of the external world, and this will be different from Anazognosia. The memory span, because you want to be sure that the patient have enough working memory to manipulate concepts, and the motor index, because you don't want the severity of the motor impairment to have an impact on your Anazognosia measure. So if you do so and you look at the localization of the lesion that will lead to this syndrome, you find at the top left damage to the insula and the putamen and a little bit of the pallidum, but mostly the core of the damage is in the white matter. So it looks like you have a lesion that is happening here systematically that will disconnect a circuit of regions that need to interact for you to be aware of your own body. And if you do the disconnectome analysis, if you look at probability of connection and you try to see how this relates to the performance, you find a network of 3 connections that are actually coming out as significant, which are like ventral right frontoparietal connection, as a cingulum, and as a pre-SMA connections. And those belong to different tracks that we investigated, and then we did some Bayesian statistics to investigate which of those predictors is the best model to demonstrate the occurrence, the incidence, of Anazognosia for hemiplegia. And we've been able to show that the insula, the temporal poles, the putamen, or any gray matter region that we reported here were significant, but really white matter tracks are better predictors, such as frontostriatal, the cingulum, the frontal ascending tract, and the ventral frontoparietal network, SLF3, are best predictors for Anazognosia for hemiplegia. But most importantly, if you combine those disconnections together, you have really the best prediction of Anazognosia for hemiplegia. So Anazognosia for hemiplegia is not coming from a brain regions. It is not coming from a single track. It is coming from a 3-parted disconnection of different systems. You have a system that is related to the update of beliefs about your body, which will be related to the mirror neuron system and the ventral frontoparietal network that is represented by the ventral frontoparietal network. The preparation of action and the update of what you're doing in terms of action, which will require as a pre-SMA, and the writing inside your memory of this change in your body and its relation with emotion that will be represented by as a disconnection of the cingulum. So what we essentially demonstrated here is that you can change your vision. You can you can use a method, radically change your observation, and this will change your theory. And so what we suggest here is that the integrative association between brain areas predict better high-level cognitive symptoms rather than isolated brain regions. That would be only possible because we changed the method of looking at things and we used connectivity approaches. We take people on the train with us, use the software, demonstrate that we're wrong. It's okay to be wrong. We provide it to you. It is free and you can download it on our website. What's interesting is that by doing so, we reconcile two fields of neuroscience, which is the research in brain lesion and symptoms and the research in functional neuroimaging in healthy controls. And by bridging the gap between the two, we are clearly strengthening the basis of our knowledge in neuroscience and helping us to move on to the next questions. And with what is interesting with that is future application of our software. You can use all of that has been done in the past 50 years. You just got to get the lesion and put in the software and see really the symptoms from a new perspective, which is just like the regions that were disconnected. And you see how by changing the method, we changed the way we observe and we changed the theory. And this is interesting, but that brings us back to our question of epistemology. What is knowledge? What is its nature? How did we obtain it? And is it really enough? Now, this question has been highlighted by two philosophers. They say that theory and models can be biased by the deceptive aspect of perception. And we've seen it by lesion analysis, classical lesion analysis. A lot of things don't come out as significant. But if you look at disconnection, suddenly you change your perception and you get something significant. And then if it seems to bring things together by default, but that opens up additional questions, such as, I just provided a method to look at disconnection and we discovered this. How much is there that we don't know about that could explain function? And like the main issue in epistemology and the main question is that an observable cannot be detected by definition and are epistemically unavailable. You cannot build theories on it. They are not part of our knowledge. So if you have your virtuous cycle here that work together to enlighten its middle what we know, it's very important to keep in mind that the size of the unknown is there and there is no way to know how big it is. And so like the only way to start to see a little bit what is not known is try to have the most comprehensive approach and see whether in this most comprehensive approach you have bias in your vision. And this opens gaps in which you can try to investigate how to fill the gap, how to circumvent the bias. So we try to do that. We try to be as big as possible. So we did our classical thing. We use lesions and we use a lot of it. We use 1,333 lesions that we converted into disconnections. And what's very important to know about brain lesions, they're not randomly distributed. So in order to control that random distribution, we created a data set of lesions that are fake, but completely randomly distributed. And each of these lesions is matched with a lesion of the true stroke lesions. And then we started looking at how do they distribute and can we model them. So if you look at the lesions, so here we have a representation where each point is in a place in 2 dimensions, and points that show a similar profile of lesions are closer together, and the ones that are the most different are further apart. And we can see here that it's a good mix. You have some overlap and some little cluster of regions that are damaged, but most of it, lesions, even if they overlap, seems to be more or less pretty unique. On the other hand, if you look at the brain disconnection, you can see that you have clear clusterization of brain disconnection, and this clusterization is even stronger in the case of stroke disconnection than it is in our control of randomly distributed lesions. If you have redundancy, if you have clustering, then you can eventually model it. So we look at the pattern of disconnection of every patient and every fake lesions, and we coded it as a pattern of disconnection in this atlas, which is a Madlasa Nature Atlas of 2016 that gives you gorgeous 360 brain areas to which we added subcortical regions because it does not involve the subcortical regions. And by doing so, we've been able to create a profile of disconnection for each patient, on which we applied principal component analysis that will give us a compression, that's a data compression, a model, if you want, of factors that will explain the disconnection. Do you have an example here from this paper in Nature from Sebastian Seung? So the idea, if you look on the left, is like you have the gray circles. The gray circle represent all the measures that you can have, and all these measures, there is some redundancy in it. Main factors that you can extract that together can reproduce your original image. It's a little bit like, MP3 is to wave sound. Like, now we compress music and you can have it like on your phone. Before it was way bigger. Thanks to this approach, now you can actually have almost the same quality of sound, but like very much compressed. Same idea, if you do it on faces, you can use different models such as non-negative matrix factorization, for example, on the top. And every faces shares similarities. We all have eyes, a mouth, a nose, and some people have a bigger nose than others. And the way this algorithm is going to work, it's going to identify similarities across participants and tell you how much of this eye or this nose do you have. You can do the same example if you had, instead of faces, 1,000 different soups, and you apply your algorithm on it. The algorithm is going to taste every soup and it's going to tell you, well, we have this main ingredient, and this main ingredient or component is carrots, another one is salt, another one is leek, and these ingredients, the algorithm can tell you how much you have in all your soup. You can have 1,000 soups, but you don't have 1,000 ingredients. You have way less ingredients. I can create all this variability of different soup. You can do the same thing with faces, and you can do the same things with brain lesions and brain disconnections. This is what we did. And if you do so, I'm sorry, I forgot to put like the caption here. if you do so, you can see that brain lesions in green, or synthetic disconnection of random lesions. You can model them, but you really get a cap at 70, 80 percent of the variance, percent of the signals that you can explain, which means you can find all the ingredient of brain lesions and brain disconnection, but together you won't have exactly the same taste. You won't create exactly the same lesion. You won't create exactly the same synthetic disconnection. On the other hand, if you take stroke disconnection, with 30 ingredient, you're already at 90%. So that means that you have the recipe of your secret soup. You can use 30 ingredients and reproduce exactly the same pattern of disconnection of your patient just by combining these components. And each of those components is a pattern of disconnection between brain regions. So we did what we did best. We compared it with pattern of activations in healthy controls. All these components, all these ingredients of brain disconnection, we compared them one by one with any profile of activation and try to see whether there is a relationship between the ingredients that make a brain disconnection, the components that make a brain disconnection, and a pattern of activation in healthy controls for a given function. And 90% of them were having a profile. 90% of them were correlating significantly with a pattern of activation related to function. And you can see here an example of 4 of those, like for example, the ingredient number 1, which is a specific profile of disconnection, correlated significantly with articularly related activation. Another one, ingredient number 6, which has a different pattern of disconnection, correlated very significantly with navigation-related activation. Another ingredient, component number 10, correlated with eye field-related activation, and component number 12 correlated with calculation-related activations. Isn't that great? It means that behind the profile of disconnection of stroke patients, you have specific ingredients that match each one of them different functions. I found that fascinating, but that made me worried. I said, why do we have such a perfect relationship between brain disconnections and brain function? And so that led me to think about, where does this knowledge come from? How do we know really about brain function? Well, we know about brain functions because a patient was coming into the doctor's office with a stroke, and it was just saying that he was not doing okay. And years after years after years, centuries after centuries, we've been asking ourselves, well, what is wrong with this patient? So our observation of what is wrong with a patient is completely led by the pattern of lesion or disconnection that occur in the patients. But what did I just say? I said that this pattern is highly biased, very repeatable. Like you always have the same pattern of brain disconnections in patients. So if that is true, I mean, the components of stroke disconnection should correlate better with task-related activations than just randomly distributed brain lesions. And this is what we've been testing. We looked really at the relationship between the pattern, like the different ingredients that make a lesion as a control, the different ingredients that make a synthetic disconnection with these lesions that are randomly distributed but match with stroke lesions. And we look at the strength of the relationship with function for the real stroke disconnections. And what we found is, yes, indeed, stroke lesions, they don't really have, the pattern of stroke lesions don't really have a direct relationship with the pattern of activation in healthy controls. The main reason being that some stroke lesions are completely in the white matter and activation are in the gray matter. So if you don't look at disconnection, these patients shouldn't have any deficits. Actually, they do. Then we compare like the synthetic disconnectome, so lesions that are not true, randomly distributed, but match with real stroke disconnectome. So relationship between its ingredient and functional activation, which is significant. And we compare whether this was as strong as the one in the stroke disconnectome, which is like true lesions. And stroke disconnectome have a tiny advantage on synthetic disconnectome. And this difference is the bias that we have in observation in stroke lesions that reflected into the fabric that we have ahead of the cognition in humans. Like the different functions that we imagine we have has been biased by the observation of stroke patients. So, this is all statistics and might seems really abstract. We thought like it'll be interesting to give it like a lot more visual and projected onto the brain. So we created maps that will represent the degree of disconnection for each of the components. And to do so, this is a high dimensional linear regressions where you regress out the loadings of each component to the probability of disconnection of each patient. We split our data set of 1,333 patients in two data set of 666 in order to assess the replicability or reproducibility of our maps, which were showing a correlation of 0.81, which is a strong effect of reproducibility. And what we can see in the next slide is the same example of the four components I was talking about before. So one that correlated with the articulatory functional activation clearly involves the arcuate fasciculus, so Broca territories, the insula, and also white matter that is behind that interconnecting. So you can see these beautiful U-shaped fibers between the Broca territory and the insula. The areas that are classically disconnected involves a classical Broca-Wernicke model plus some extra regions in the intraparietal sulcus. The sixth component that was correlated with navigation disconnect the parahippocampal gyrus from the retrosplenial cortex and involves the posterior branch of the cingulum. We can also see some disconnection from the visual afferents that are really important for really navigating yourself in space. As a component number 10, that was correlating a lot with functional activation in healthy controls during movement of the eye field, involved the frontal eye field bilaterally. You can see an asymmetry where on one side it is split, on the other it's all together. This has been a finding from Matt Glassat before. And you can also see that they're highly interconnected by the corpus callosum. And that does make sense, because if you had a lesion in the frontal area field, the white matter subjects into the frontal area field, you have a neuropsychological syndrome of deviation of your gaze, looking to the side of your lesions, and your two eyes actually deviate. And here we can see that they're highly interconnected and they come together, and that would be the explanation why the two eyes, not only one eye, deviates. Finally, you have the calculation that component number 12, calculation, correlated with calculation. And you can see here that the component is a single region with its subjacent white matter. And we do know, because it's some papers have been written by Andreas Kleinschmidt on the Guessman Syndrome in 2011, that this syndrome is due to lesions that disconnect short U-shaped fibers within the region of an intraparietal sulcus in the right hemisphere, leading to acalculia, agraphia, left-right disorientation, and digital agnosia. In the left hemisphere, sorry, I said right hemisphere. It's in the left hemisphere. Then we were thinking, oh, that's great. And so we have like 40 components, and we have 40 correlations with functions. But I just showed it to you before that actually some function are emerge from the integration between other functions. Typically category fluency, you've seen that it is emerging from the interaction between semantic executive function and the production of language. So one would be the same here with our components. So what we did essentially is try to see how much each component was correlated to each functional MRI term of the meta-analysis, to try to predict with it the functional MRI term and extract from this prediction a map that will show us the white matter that is behind each of the term of neurosynth. And we did it twice again with our two data set of 666 patients and shows that they were highly replicable a second time. And by doing so, that led us to have 590 maps of the white matter supporting 590 terms, selected terms of the neurosynth database. So, you know, I just want to reassure you that I'm not going to show you 590 maps right now. That's not my purpose here. What I'm going to do is I'm going to show you a map that show the top contribution of the white matter to those terms, rather than showing you all the terms. And then I'll show you how those terms are reliable in terms of effect size, and also how many terms loaded per regions. So this is what we have. This is just one slice of white matter. You can see the major forceps contributing to nouns and the minor forceps contributing to competing and fluency. Middle of the brain, where you have the phonics, you have a lot of memory and association with the context. And then on the left hemisphere, you have a lot of language with verb, fluency, pseudo-words, syntactic. A lot of, you have a bit of phonological. And as you're going further in the frontal lobe, you have more sentences, memory tasks, decision making. And if you're going more posteriorly, it's all about more written words and noun and all these kind of things. On the right hemisphere, it's more about navigation, the tone, and maintaining informations. This is really the top contribution, the most significant relationship with the white matter. It is a high dimensional map, as you might imagine, with 590 dimensions. We repeated that for several slides in the brain. The next question that we had really is how strong is this result? So we looked at what we call the effect size. The effect size indicates to you how much of the variance of the task-related activation is explained by your model. So here we have R value, which is a test of fitness of the model, and you can see that you have a pretty high effect size because we are above 0.5 anyway, and we reach in some places 0.8, and that's actually pretty good. With of course, we have some areas that are not well covered, which are mostly part of the brainstem and the cerebellum that are not yet perfectly understood in comparison to the rest of the brain, of course. But you get what I mean. And then we were like, okay, this is interesting, and this is a top map, but how many terms with a good effect size are loading for each part of the brain? And here we found that there was a strong asymmetry, because in the left hemisphere, you have up to 70 different terms that can load onto white matter tracks, while in the right hemisphere, you barely have one or two. And this demonstrates clearly the asymmetry that we have between what we know in terms of function about the left hemisphere and what we know in terms of function about the right hemisphere, which is very much limited. And so that gives you, again, by looking at it from the whole perspective, that gives you an extent of the huge gap in knowledge that we have here that you can feel in your generation and the next generation of the people you will teach. This method that we provide with a component allow you for any pattern of activation. Here you have an example of the right figure tapping and the left figure tapping to derive the profile of disconnection, of significant disconnections in patients with stroke that will explain this pattern activation, and then identify really the lesions that are behind this pattern of disconnection. This data set was provided by my colleague Laurent Petit, that you can see here at the bottom right, from the team Gene that I'm head of now. So in terms of conclusion, for this lecture, well, we showed that the redundancy in brain disconnection after stroke shows striking correspondence with task-related ephemeral activation patterns. Sharpen your criticism. It is weird to have such a strong relationship, and that means maybe that there been some bias in the way we explore functions, mainly due to the pattern of disconnection in stroke. This correspondence is due to the influence of the organization of white matter connection on the functional segregation of the human brain. And it is also due to the biased distribution of brain lesions that has also biased our taxonomy of brain functions. And the two together give us the opportunity to build an atlas of function as we know it, because we use the current state in the art of task fMRI and white matter connection mapping to build the first atlas of the function of white matter, knowing that we don't know all the functions, we don't know all the connections, but this is good enough to build an atlas of what we know. It is important to know that the functions that are showed in the atlas in the white matter correspond to the joint contribution of the areas that are connected by this white matter. And in sum, while our method allows us to map for the first time white matter function and may help predict patient syndrome, the biased report, which is an epistemological bias, provoke questioning and further exploration of the definition of brain regions, particularly as we've seen for the right hemisphere, where very, very little terms were loading. All right, thank you for your attention, and we'll see each other again for the next recorded lecture that will be about brain hierarchies, which is very important for USA this year. Brain hierarchies are actually regions that are working together to build incrementatively the function as we know it. So it is very much into the associationist perspective. I want to stress that the 12th and 30th November, we have a special issue symposium on structural connectivity of the cerebral cortex. You are invited to watch the video that will be uploaded on YouTube. And then every Monday morning from 9:30 to 10:30 a.m., we do the Neurochino, grab your coffee, and eventually, if you've seen an interesting paper from the last week, and join us for the discussion of the exciting science that happened. It is all about being curious and excited about science, and that's what the secret behind making great discoveries. It is from 9:30 to 10:30 a.m. In terms of evaluation, and I will never repeat that enough, the scores that you will have to the principle of cognitive neuroscience module will be averaged between the part A and the part B, which is led by Professor Valesi. So you'll have a test for part B with Professor Valesi, a test like that I'm going to explain with me, and this score will be the score to part A and part B will be averaged. That will be your total score to this module. The way I score people is related to a combination of your attendance to the different events that I organize to train you as future researchers, your participation. So it cannot be the participation really in class because this is recorded due to the current sanitary situation, but you can leave comments and questions on YouTube, that's all on Neurostar, and this is really important that you start being curious and ask questions and being critical. This is part of your job. You need to bulk up your brain and start thinking about those lectures instead of passively listening. And then every year, as a student to write an essay. This is a little project. This year, I want you to write this essay about a brain hierarchy, the one of your choice. You have several different hierarchies that exist in the brain. Try to be original. This essay is organized in 5 sections, which in total will be 1,500 to 2,500 words, not including the reference at the end of your document. And they will combine a general introduction that needs to be captivating even for people that are not from the field of neuroscience or cognitive neuroscience or neuropsychology. In other words, your friend, your mom, your parents, they need to be interested about your essay. Make me excited about this, really. Another paragraph, which is about the anatomy of this hierarchy. So what are the regions? Why is it a hierarchy? What really is the order behind this hierarchy in the brain? Then another little paragraph about whether this is lateralized or not. Sometimes the hierarchies that you chose has never been explored in terms of lateralization. And this is very interesting. This is where you can be original and suggest how would you really measure this lateralization of this hierarchy in the brain. Then another paragraph about the link between this hierarchy and brain evolution. A lot of models that are showing that we add new level of in hierarchical processing with evolution. Just build it up. You do a lot of paragraph about this, and then a synthesis where you show how everything works together. A nice conclusion where like you show that it all makes sense. All this in 1,500 to 2,500 words max. I want this document sent to me by email, to my personal email, michel.thiebaut@gmail.com, before December 11th, midnight. Okay, this is the deadline. If you arrive on the 12th, this is too late. December 11th, I want this document in my email. Okay. Well, I thank you very much for your attention, and I look forward to see you on Zoom during the NeuroChino and during the practical sessions. See you soon. Bye.